Hello, wonderful people. Here we are back. This is chapter 4, and we're going to be covering a little bit about microscopy and staining. This should be a little bit easy because it correlates very nicely with what we did in the lab this week. So hopefully you've gone through the PowerPoints and you've learned a little bit about microscopic morphology and arrangement and staining procedures. Okay, so that's 2/3 of what this lecture is about. The last third is about microscopes. So this should flow very, very nicely. Okay, we're going to start talking about the different types of microscopes. Okay, the bright field microscope. The bright field microscope is what you and I typically use in the laboratory as a bright background, and you can only look at things that are either colored, naturally colored, or are dyed. We've killed them and we've stained them the way that we have done in the simple and differential stains. Okay, so we're looking at killed and stained specimens. Okay. What else do we know? It takes a series of lenses for magnification. On a typical microscope, for microbiology, we use an objective that is 100x, and we use an ocular that's 10x. What does that mean? Well, if we look at a microscope here, okay, here's our objectives down here, and here's our oculars. So the objectives are those lenses that are close to the object, and the oculars are the lenses that are close to the ocula or the eye. Okay, so again, in microbiology, we usually use a 100x objective, and the oculars are usually 10x, and the total magnification is 100 times 10, which is 1,000x. So that's 1,000 times magnification. We usually use oil immersion. So what does that mean? That means that you have your slide, and you add a little bit of oil. See the oil here? So you can see here, this slide allows you to contrast what's happening in the objective in the absence of the immersion oil versus the presence of the immersion oil. So here you can see that there's at most about 3 rays coming from the specimen. By contrast, here we have about five different rays coming from the specimen. So what happens is that more light information makes it into the objective. So interestingly, the immersion oil actually makes the image slightly smaller because it's pulling all those light rays in. But because it brings so much more information into the objective, the resolution and the contrast in of the subject is much higher, and so that's why we use immersion oil in that 100x lens. Okay, let's see. So you have your low slide. You put 1 or 2 drops of immersion oil. You immerse your highest magnification objective in there, and then that allows you to see your object. Okay, here's the next type of microscope. It's called a dark field microscope, and in a dark field microscope we have what's called a dark stop. And in the dark stop, what it does is it prevents the main light coming from the specimen from entering the objective. So only the rays that are scattered by the specimen enter it. Everything inside of it does not make it into the objective. Okay, this is desirable when you need a high contrast image because you're looking at something that is very pale and because it's alive. Okay, now I am not fond of dark field microscopes. They do have very high contrast, and you're able to resolve a lot of things. But this is what it would look like. This upper right-hand corner one. Okay. So again, here's your bright field, only stained organisms could be seen here. Here's a dark field. It doesn't need to be stained. These could be things that are alive that you can see in here. Very high contrast. You can see these little white images here that if they're inside, they're mitochondria. If they're outside, they're bacteria. So very good contrast. But I'm not fond of this dark background in the dark field microscopes. Whoops! I just got rid of my. Let's see, we can do this again. There we go. All right. So, pale objects, and consequently objects that are alive, okay? Only those rays that are scattered by the specimen enter the objective as that net dark background, and the resolution and contrast is very, very high in dark field. Next comes phase microscopes, okay? So in phase microscopes, instead of having a dark stop at the bottom, what it has is a quartz dial of different thicknesses, okay? And you know what? I'm going to stop this for a second because there's something I want to show you. So I'm going to see if I can. Okay. So hopefully this works, okay? This is an image that I borrowed from the internet, and it's okay. It's from the same author that we have right now, okay? And it shows how a phase contrast microscope works. So again, instead of having that dark stop like in the dark microscope, this phase contrast has this thing called a faceplate. And this faceplate is a piece of glass of varying thicknesses. You can literally turn it, and a different thickness gets associated with the incoming light. So imagine that here's your low bacteria, okay? And there's a light source coming over here, all right? And when the light source interacts with the bacteria, it causes one type of a wavelength that goes through one part of the faceplate that doesn't change in thickness. However, a second ray of light does in fact enter the faceplate, where as you rotate it, the thickness of the faceplate changes. And what this does is it causes the two rays, the wavelengths of the two rays, to either come together in such a way that they add or in such a way that they subtract. And to illustrate that, I have this little guy right here, okay? This is a sine wave, kind of the way that light is a sine wave, okay? And it looks like I only have one guy right here, but it's actually two. It's an orange one and a blue that's right underneath it. And because they're both right on top of each other, we have this sum, which is this gray one, which is really, really large. Well, if you have that, if you have two waves where the peaks and the troughs are just perfectly aligned, they add to each other to make a higher intensity wave, and you end up with a bright spot in your microscope. But as you move this faceplate around, okay, and I can do that mathematically over here by moving one of the well, I thought I could. Let's see. Okay, let's see if it works now, okay? So I'm going to change that increment a little bit. And notice that now we can see that there's an orange and a blue wave there. If I change this some more, to get further apart, here's at 0.5. And look at what's happening to the gray one. As these get further and further apart, the gray one becomes smaller and smaller. So as the troughs and the peaks of these two waves move away from each other, the sum becomes smaller and smaller and smaller, okay? Let's go to 1 here, see what happens. Okay, then made it smaller, 1.5, okay? There's a sweet spot, okay? There's a sweet spot where one wave can be exactly the opposite of the other. See how the orange is right here and the blue is over here? You have a peak with a trough and a peak with a trough, and they're exactly subtracting each other so that the sum is this great bar right in the middle, okay? When that happens, the one wave subtracts from each other, and that negative comes out as a dark spot, okay? So there you have it. As a wave sum, okay, you either end up with a bright spot when they're aligned with each other, or you come up with a dark spot when they're the opposite of each other. And that is controlled by moving this faceplate around, okay? So what does that look like? Well, it looks like this. Here's a phase contrast. Notice that it has not only does it have high contrast, there's actually a three-dimensional aspect to it. It is slightly three-dimensional. And again, the interesting part about it is that if you don't like the way that it's contrasted, you just take that phase contrast and you move it a little bit more, and different parts of the cell contrast. So that's very beautiful, okay? So that's phase microscopes to observe the internal structures of living microbes, okay? So you can use them with live specimens. And are we done? No, we can keep on going. The next one is what's called a differential interference contrast. Same principle as this phase contrast, except that instead of having that so-called faceplate, there's one there's not only a faceplate, but there's also another filter in the objective, and they work synergistically to provide a very three-dimensional cell. So this differential interference contrast, or so-called Domarski, is one of the best ways to view cells. And if you have to manipulate the cell, if you have to put something in it or take the nucleus out or whatever, this is the only microscope that will allow you to do that. Okay, so very neat stuff that takes place here in microscopy. What would it take to see a living specimen? Okay, well, the choice would be phase contrast. What would it take to be able to see a flagella, for example? The choice would be a phase contrast. What would it take to see a mitochondria in a living organism? The answer is phase contrast. Why are we not saying differential? Differential would work, but these are very expensive. So typically, if we're looking for something that's alive, this is lowbrow, this is too expensive, we go to phase contrast. So there's your answer to your question. If you're looking at a live organism, you're going to be using phase contrast. If you see a picture in the background is dark like this, that's a dark field. Okay. All right. Are we done? No. Now we move on to a very exciting piece called fluorescent microscopes. Okay. And the reason that these are interesting is because we're adding this tool called immunofluorescence to a microscope that uses UV light. Okay. So here's a light microscope of a specimen. Here's a light microscope using immunofluorescence. Okay. And notice that the bacteria are, I mean, they're very well defined. Okay. But that's not the exciting part about immunofluorescence. The exciting part about immunofluorescence is that, as the name implies, it takes antibodies. Okay. If you don't know this or don't remember this, antibodies are very, very specific. They only claim to very specific molecules. Okay. So that's a hallmark of antibodies, is their incredible specificity. Now we add this little thing called a fluorescent dye, which fluoresces in the presence of long-wave ultraviolet light. Okay, and the reason that this is important is because if the antibody adheres to something, we know what it is. And the reason that we know what it is because that antibody will only adhere to one specific organism, one specific structure. So if it's sticking to something, we know what it is. And consequently, immunofluorescence is diagnostic. We actually know the identity of the microorganism when it decides to fluoresce. Okay, so that's what's incredible about this. Okay, we can see local specific chemicals or structures within cells. Okay, and it is a quick diagnostic tool. This is probably the only diagnostic tool from a microscopy point of view. Very incredible stuff. Okay. Are we done? No, we can keep on going further. Now we have this thing called a confocal microscope. Okay. Some of you may understand what I'm talking about here, because what I'm going to do is I'm going to take you back to the way that cameras used to be used, film cameras. And those film cameras used to have what's called a depth of field. And the smaller the aperture, the greater the depth of field was, which means you could see things in focus far behind and far in front. Confocal microscopes use what's called a they use a pinhole aperture, so that as much material can be in focus as possible. Okay, and I'm going to stop right now for now, and I'll come right back in a moment. Okay.